For today's discussion, our topic is biosafety and risk assessment. Hello there, class. Meet the members of Group 9. Christine May Gomez, Claire Ann Santelian, and truly yours, Shana Valerie Maratas. By the end of this lesson, students will be able to explain the importance and the significance of biosafety and risk assessment appropriately in responding to community disaster emergencies. The scope of this lesson are the biosafety and risk assessment, risk analysis, assessment management, and also communication, the biosafety levels, and also its elements of biosystem, and also the Cartagena Protocol, risk groups, and lastly the NIH guideline definition and the risk groups. So first, let's talk about what is biosafety. So biosafety is used to describe efforts to reduce and eliminate the potential risk resulting from biotechnology and its products. It has similarly been defined as the avoidance of risk to human health and safety, and to the conservation of the environment as a result of the use of research and commerce of infectious or genetically modified organisms. So this is really relevant in scientific disciplines that underpin biosafety studies include molecular biology, plants breeding, and genetics, plant pathology, also agronomy, weed science, and also entomology and ecology, among others. Adopted during the United Nations Conference on the Environment and Development, addresses the pressing problems of today and also aims to preparing the world for the challenges of the next century. So it reflects a global consensus on political commitment at the highest level on development and also environment cooperation. This is actually a successful implementation is at first and foremost the responsibility of the governments. Biosafety emerged as a global priority in Chapter 16 of Agenda 21 and in Articles 8 and 19 of the Convention on Biological Diversity. Risk analysis must be undertaken to predict the occurrences of negative impacts on the environment and human health. The backbone of the practice of biosafety is risk assessment. And risk assessment should be performed by the individuals mostly familiar with the specific characteristics of organisms being considered for use, also the equipment and procedures to be employed, animal models that can be used, and also the containment equipment and facilities available. As we all know, risk is defined as the probability of harm. On the other hand, risk analysis is a multi-step process aimed at mitigating the impact of risk on business operations. Leaders from different industries use risk analysis to ensure that all aspects of the business are protected from potential threats. Performing regular risk analysis also minimizes the vulnerability of the business to unexpected events. Risk analysis follows a structured approach with three distinct but closely related topics, or the steps. First is the risk assessment. Next, we have risk management, and also lastly, the risk communication. Risk assessment following Codex Alimentarius noted in the Food and Agriculture Organization's Biotechnology Glossary. Risk assessment is defined as scientifically based process consisting of the following steps. The first is the hazard identification, second, the hazard characterization, third, exposure assessment, and also the last fourth is risk characterization. We have this first example: Health and safety risk assessment. So this is a type of risk assessment used by the safety managers to determine health and safety risks associated with the job, work environment, and the current processes. We have this another example, the workplace risk assessment, which is performed by office managers and school administrators. This is actually a tool that helps ensure that a workplace is free from health and safety threats. The third example is the fall risk assessment. So this is performed by the nursing staff or aged care units or the centers to evaluate the possibility of falling. The last example we have construction risk assessment. So this is a vital assessment used in the construction site to help safety teams implement corrective measures and stakeholders comply with safety regulations. Now let's move on to risk management. So this is a process distinct from risk assessment, of which weighing policy alternatives in consultation with all interested parties, considering risk assessment and other factors relevant for the health protection of consumers and for the promotion of fair trade practice. And if needed, selecting appropriate prevention and control is an option. Let's take a look at this example. Risk management strategies include leveraging existing frameworks and best practices, minimum viable product development, contingency planning, root cause analysis, and lessons learned, built-in buffers, risk-reward analysis, and also the third-party risk assessments. And now let's talk about the risk communication. So risk communication is actually defined as the interactive exchange of information and opinions throughout the risk analysis process concerning the risk-related factors, and risk perception among risk assessors, risk managers, consumers, industry, and the academic community, and other interested parties, including the explanation of risk assessment findings and the basis of risk management decision. So this risk communication involves an ongoing dialogue between regulators and the public about risk and options to manage risk so that appropriately decision can be made. Risk assessment also should be carried out on case-by-case basis. There are several ways to complete a risk analysis, as according to Connor et al. Of 2003. It has been generally accepted that details of risk assessment procedures may vary from case to case, but there are few logical steps that need to be followed. First, identification of potential adverse effects on human health or environment. Second is an estimation of likelihood of these adverse effects. Third is an evaluation of identified risk. Then, considerations of appropriate risk management strategies. And then the assessment of overall potential environment impact and consequences. Lastly is a recommendation as to whether or not the risks are acceptable or manageable.
Biosafety level. A biosafety level is the level of the biocontainment precaution required to isolate dangerous biological agent in an enclosed facility. There are 4 BSL. The primary risks that determine levels of containment are infectivity, severity of disease, transmissibility, and the nature of the work conducted. Biosafety Level 1. It is suitable for work involving well-characterized agent not known to cause disease in healthy adult human or individuals, and of minimal potential hazard to laboratory personnel and the environment. Biosafety Level 2. Risk Group 2 infection agents are pathogens that can cause human or animal disease, but under normal circumstances are unlikely to be serious hazard to laboratory workers, the community, livestock, or the environment. Level 2 infections are not considered to be a serious hazard. They are a moderate individual risk and limited community risk. Biosafety number 3. Risk group 3 infections agents are pathogens that usually serious human or animal disease or which can result in serious economic consequences but do not ordinarily spread by casual contact from one individual to another. High individual risk or low community risk or that can be treated by antimicrobial or antiparasitic agents. The last biosafety level 4. Highly toxic or infectious agents that are at a very high risk for forming infection aerosols. So, here are the elements of biosafety system. First is the guidelines formulated should be transparent, science-based, and flexible. The guidelines clearly define the structure of the biosafety system, the rules and responsibilities of those involved, and how the review process is to operate. Second, competence and confidence must be built in the people who are involved. The people are knowledgeable and well trained, confident in their ability to make decisions, and supported by their institutions. Third, the biosafety review process must be appropriate. The review process is based on up-to-date scientific information, focuses on specific combination of crop, gene, and environment, promotes appropriate risk management practices, and balances risk against benefits. And the last one is there should be means of ensuring feedback so that the system improves through experience. Feedback mechanisms are used to incorporate new information and revise the system as needed.
Biosafety and Cartagena Protocol. The Cartagena Protocol on Biosafety is an international regulatory framework established under the Convention on Biological Diversity or CBD. It focuses on ensuring the safe transfer, handling, and use of living modified organisms or LMOs, with a particular emphasis on transboundary movement. So the protocol primarily addresses LMOs intentionally introduced into the environment and genetically modified farm commodities. It does not cover pharmaceuticals for humans or products derived from LMOs, such as cooking oil. The RCGM, established under the Department of Biotechnology or DBT, supervises research activities including small-scale field trials, whereas approvals for large-scale releases and commercialization of GMOs are given by the GEAC, established under the MOEF. The SBCCs and DLCs have a major role in monitoring. India ratified the protocol in January 2003, with the Ministry of Environment and Forest being responsible of its implementation. The Ministry of Environment and Forest has undertaken various initiatives to fulfill its obligations, including capacity building for effective implementation. To support this, the Ministry of Environment and Forest is implementing a capacity building project on biosafety, funded by the Global Environment Facility or GEF and the World Bank. The project aims to strengthen the regularity framework, enhance risk assessment and management, provide training and human resource development, and facilitate information sharing related to the transboundary movement of LMOs and GMOs. In summary, the Cartagena Protocol on Biosafety provides a regulatory system for the safe handling of LMOs, and India has ratified the protocol, with the Ministry of Environment and Forest leading efforts to implement it through capacity building initiatives and projects. Risk groups. Infection agents or pathogens are disease-causing microorganisms. There are hundreds of microorganisms that cause disease in humans and animals. They can be classified as bacteria, fungi, viruses, prions, or parasites. The principal hazardous characteristics of an agent are its capability to infect and cause disease in a susceptible human, animal, or plant host, its virulence as measured by the severity of disease, and the availability of preventive measures and effective treatments for the disease. Other hazardous characteristics of an agent include probable routes of transmission of laboratory infection, infective dose, stability in the environment, host rate, and endemic nature. Each infections agent or microorganisms can be categorized by four levels of hazard known as risk groups. It is important to keep in mind that in many instances, a risk group will correlate with the biosafety level. RG2 agent will be handled at BSL2, but they are not equivalent to some RG3 agents may be safety manipulated at the BSL2 under certain conditions. NIH guidelines definition of risk groups. Risk group 1 or RG1. Agents that are not associated with disease in healthy adult humans. This group includes a list of animal viral etiologic agents in common use. The agent represents no or little risk to an individual and minimum risk to the community. Risk Group 2 or RG2. Agents that are associated with the human disease, which is rarely serious, for which preventative or therapeutic interventions are often available. These agents represent moderate risk to an individual but a low risk to the community. Risk Group 3 or RG3. Agents that are associated with serious or lethal human disease, for which preventative or therapeutic interventions may be available. These agents represent a high risk to an individual and a low to moderate risk to the community depending on the pathogen. Risk Group 4 or RG4. Agents. The exotic agents that are likely to cause serious or lethal human disease, for which preventative or therapeutic interventions are not usually available. The agents represent a high risk to an individual and a high risk to the community.